Okay. Good evening, Manvita. Good evening, Shani, Viva, and good evening, Gopika. Okay, so good evening, guys. We can start the class, right? Probably, I think I'm live. So YouTube live. I'm live anyway. I'm also living, and YouTube live is also going on. Okay, so quickly, I will also start the recording. Uh, Really? No, I think prokaryote different photo is there, right? I'm not sure, guys. Probably I thought that prokaryotic different photo is there. It's a eukaryotic cell. You can see, right? It have organelle and all the things. Prokaryote, I'm not sure. I will check. Okay, sorry. Okay, guys. So we will come to the sorry. We will solve some question. Good evening, Shreya. So first question: nuclear matter without envelope occurs in. They're asking where you will get a nuclear matter without an envelope. Okay, the options are bacteria and green algae, cyanobacteria and red algae, mycoplasma and green algae, cyanobacteria and eubacteria. Okay, think about it, and I will on the poll. So, okay, I'm launching the poll. Who are in the Zoom, try to answer in the Zoom polls, and who are in the YouTube, try to answer in the YouTube live chat. Good evening, Aditi. So I'm just waiting for your answers. Yeah, so many people gave the correct answers. Still, some of you are like thinking. I think, yeah, easy question. If you know the those organisms, it's like easy, dead easy. They call it dead easy, right? Like that. I don't know why they call it dead easy. Like I'm not sure. Are they saying like easy like death? Like I don't know. We'll see. I'll try to find the answer. Okay. So they're asking nuclear matter without envelope occurs in. Okay. You guys have last 10 seconds. So I'm ending the poll. Okay. I'm ending the poll now. Oh, like that. Then that will be correct answer. Like dead person can also answer. I'm sharing the results. 27% chosen option A, 9% B, 13% C, and 51% D. Okay, I'm stop sharing the result as you guys are correct. Okay, so first thing, and in YouTube I got like one option, sorry, two option as a D. Good evening, Ashwin. So nuclear matter without envelope. That means there should be no nuclear envelope. Where it will occur? It will occur in the prokaryote. We know that, right? We know that it's occur in the prokaryote. Good evening, Darshan. So then our next issue is like which one is prokaryote, right? Bacteria is prokaryote, but Greek algae is the eukaryote. Cyanobacteria is a prokaryote, but red algae is the eukaryote. Mycoplasma is a prokaryote, but green algae is again eukaryote, right? Here, cyanobacteria is the prokaryote, and eubacteria means the group of bacteria, or you can call it like this new or true bacteria. These are also prokaryotes, right? So the correct answer is the option D. D is the correct answer, right? Easy question. So just you have to think about the different what you can say organisms and where they belongs to. That much only the work you had, right? So we will go to the second question. I think everyone understood that one. This question is, which of the following is not a characteristic of the cytosol? Okay, I will explain what is everything. So first you try to answer, like try to find the answer. Okay, good evening, Shushma. So which of the following is not a character of the cytosol? Not, remember, okay. It surrounds the cytoskeleton. Second one, it's the fluid portion of the cytoplasm. Third one, it's located inside the nucleus and organelles. Fourth option, it contains both organic and inorganic substances. Okay, I will launch the poll. So like I'll give you 5 seconds to think. So you guys can think in that time. Okay, then I relaunched the poll. Who are in the Zoom can answer in this one poll. Who are in the YouTube can answer in the YouTube live chat. I'm just waiting. Ashwin, what happened to your DP? It's completely like black something. Okay, I'm getting a mixed kind of the answer. Okay, we'll see. We'll see what is the meaning of the question and what will be the answer and everything. Good question, Pawan. First, we'll answer that. Okay. Is any bell rings when you heard the term like cytosol? A similar kind of term, if you heard anywhere. Mohan, think one more time. Okay, I'm ending the poll in like 5 seconds. See, if you don't know the answer, no issue. Okay, if you don't understand the question, also no problem. Okay. Anyway, we are solving this. Okay, I'm ending the poll then, and I'm sharing the results. I got one like 6% A, 27% B, 48% C, 19% D. In YouTube is option. No, like nobody's chosen option. I'm stop sharing the results. Okay, so we'll see what's the correct answer. Before we going to think about the particular answer, we have to know what's the meaning of the cytosol. Do anybody know what is the meaning of the cytosol? Meaning of the cytosol. What may be the meaning? Okay. So somebody is asking, somebody's like, I'm just waiting. Anybody? Okay, I got answer from Ashwin. Apart from that, how did you guys choose the answer? Exactly. Everyone know about the cytoplasm, right? What is cytoplasm then? You guys can answer. So there is a fluid which is present in the cell, inside the cell or inside the plasma membrane. We call it as the cytoplasm, right? No, it's like partially true. What is the meaning of cytoplasm? Okay. So meaning of cytoplasm is the fluid from the plasma membrane to nuclear membrane in the eukaryote, or total membrane in the prokaryote. Okay, so that's what total fluid, you can call it as the cytoplasm. Okay, yeah, cytoplasm I'm telling. Okay, the fluid which is filled inside the plasma membrane or inside the cell and up to the nuclear membrane. Okay, up to the nuclear membrane. I will write it down this one because you may misunderstand the abbreviation. Nuclear membrane, up from the plasma membrane to the nuclear membrane. That's what the cytoplasm means. So then question, what is cytosol? Okay, this is very simple if you understand what is cytoplasm. Cytoplasm without cell organelle, without organelle, fluid inside nuclear membrane, that's different. We'll come to that. Okay, so remember the up to the I will write a cell structure here. Okay, think that so think something like that. Okay, so here to here, I think that this is the plasma membrane and this is the nuclear membrane. Okay, all the fluid which is filled in this portion, okay, all the fluid which is filled in this portion is called the cytoplasm. Okay, what is the fluid which is filled inside to that? The fluid which is filled inside to that, which is inside the nuclear membrane? Simple, nucleoplasm or nuclear plasma. Okay, nucleoplasm. Okay, so nuclear plasma or nuclear plasma. So only water, salt, mineral, 1 minute, I didn't understand. Okay, exactly. So inside the nuclear membrane is nucleoplasma, and outside the nuclear membrane, but inside the plasma membrane is the cytoplasma. But cytoplasma, if don't contain any organelle, that means if you remove the cell organelles, or as Shalini told, the aqueous component of the cytoplasm, you can call it as a cytosol. This is similar to our blood. Okay, blood. What is blood plasma? Blood plasma is the aqueous component of the blood. Or I can say in another way, right? If you remove all cellular structures from the blood, then it will be plasma, right? Exactly similar structure. Okay. So good evening, Akhil. So options are, it surrounds the cytoskeleton. No, it's a wrong, because cytoskeleton will be present throughout the cell outside, right? It's a fluid portion of this cytoplasm. Okay. And third, it is located inside the nucleus and organelle. It contains both organic and inorganic substances. They're asking which of the following is not a characteristic of the cytosol, right? So which one is a not? The correct answer is, it is located inside the nucleus and organelle. It is the wrong answer. Why it is wrong? Because it is not present inside the nucleus, but it is present outside the nucleus. So the wrong wrong sentence, or the correct answer is option C. Okay. So only what salt? Like you are asking about like cytosol only have like what salt, or water, salt, and mineral you are asking. It also contains some other organic things also, like amino acids, everything, but it do not have organelles, like it do not have mitochondria, ribosome, everything. Like you can remove those things. Okay. A option, why it is not present? It surrounds the cytoskeleton, they are saying. Okay. Remember, the cytoskeleton will be like this. Okay. It will be completely surrounding the cell. So if it means outside of the skeleton, so it means outside of the plasma membrane. Okay. Outside of the plasma membrane. But it will be present inside of the plasma membrane, right? So that's why A option is not correct. Okay. So any more doubts, guys, regarding this question? Remember, cytoplasm, cytosol, nucleoplasm. Okay. And another one thing is that it's a protoplasm. Okay. What is protoplasm or protoplast? Protoplast means a cell without a cell wall. Okay. So if you take a plant cell, a cell without a cell wall is the protoplasm. If it's animal cell, it's a protoplast. Sorry, not plasm, protoplast. Okay. What is protoplasm? Protoplasm means all together, all cytoplasm and nucleoplasm all together, you can call it as the protoplasm. Okay. Cytoplasm minus cell organelle is equal to cytosol. Exactly, Chirant. Okay. Okay, then we will move on. We are actually discussing about the Golgi complex, right? So, and we ended in here only. Golgi apparatus is the important organelle in our, in this cell structure. So where it is present then? First location, it normally present next to the endoplasmic reticulum. Next to endoplasmic reticulum. It's normally situated after the endoplasmic reticulum. Okay. Then what happens after the end? What's its function? It have two phases. One is the cis phase, another one is the trans phase. Right. One will be a cis phase, as you can see here, this side, and another one will be the trans phase. Okay. Trans phase will be the opposite kind of the phase. Okay. So as you can observe in the other thing also. Okay. One second. If you observe, it will be having different kinds of the structures. What kind of structure it will have? Can anybody answer? So one side will be, what kind of structure a cis phase will have? Cis phase will have. Exactly. Okay, I'm just curved kind of structure. Okay, concave structure. Anything else? Flat? No, not flat. As you can see here, it will be having a concave structure. Convex or concave? Convex or concave? Somebody is answering concave and somebody is answering convex. One second, guys. I'm just facing problem in YouTube. Okay. Correct. Sorry. Sir, concave lens and concave mirror are like structures. Convex mirror, like first you have to tell me like what is concave and what is like, I think you know about what is concave and what is convex, right? So space will be the convex kind of structures. I just forgot it, like so where is the space? Space means it should be a convex kind of a structure, and there is another one face. Okay, so that face is called as concave face, or we can simply call it as the trans face. Okay, concave or trans face. Yeah. So like normally how I remember, really, like you guys just missed it or I'm missing it, like so I'm getting completely like ulta kind of the answer. Hmm? Yes, concave trans, and yes, convex face only, like somebody say told that like it's in the book, it's given ulta. No, it's correct only in book also, it's given like concave trans and convex face only. Okay, so it's given like that only. Okay, like it have a two faces, we can say. Okay, so this convex phase, or actually you can also call it as maturing phase. Okay, so and this one, this phase, you can also call it as the initial phase. Okay, so convex phase you can call it as the immature phase or initiating phase. Okay, initiating phase, and the concave phase you can call it as the maturing phase. Maturing phase. Why we call in all this way, and what's the reason behind that? As you can see here, this particular Golgi apparatus will attach to the endoplasmic reticulum, and endoplasmic reticulum will send its concentric. Concentric means, as you can see, it will normally looks like this, right? So if you observe like this, it's somewhat observe like it looks like this. Okay, so that's why they given the concentric, I think. Okay, so what happens? They will enter through the space. What are the proteins which is created in the endoplasmic reticulum? Directly enter through the tubules to the Golgi apparatus. In the Golgi apparatus, some modification will happen to the protein. Okay, what are those modifications then? The modifications are mainly the addition of the saccharides. Okay, and that's what we call it glycosylation. Okay, addition of the saccharide to that particular protein. Okay, this is also helping in the attachment of the lipids. Okay, that's, we can simply call it as the production of or synthesizing of glycoprotein and glycolipid. Okay, remember, wherever you come across with this glyco, okay, that means sugar or the saccharide is attached to that particular one. Okay, a protein which attached to a sugar or the saccharide is called glycoprotein, and lipid which is attached to a sugar or saccharide is called glycolipids. Okay, like an onion, concentric harmonisha. Exactly. Okay, so synthesis of glycoprotein and glycolipids are happening here. Okay, and another one important function the Golgi have. What is that function? This is act like a carrier service. Okay, carrier service of the cell. So what is the meaning of carrier service of the cell then? So it will take the particular protein and it will put that in a vesicles. These vesicles serve or transfer the material of that particular synthesized material to the different places. It may be mitochondria, or it may be plasma membrane, or it may be chloroplast, and all the things. Okay, so what are the materials it needed then? Remember, whatever the organelle which is present in our cell, okay, not all of them have the ability to produce their own constituents. Okay, if I give you an example, plasma membrane is made up of the Yes, glycosylation occurs in both endoplasmic reticulum and Golgi apparatus. What is happening in the endoplasmic reticulum is the initial level of the glycolization, but in the Golgi level, it will be like a complete conversion or changes will happen. Okay. I will take a plasma membrane, guys. Plasma membrane is made up of the lipid and protein, right? So how the lipids are created? If plasma membrane want to grow big because cell is growing big, is plasma membrane have the ability to produce its own protein? No, it's not possible. Okay, then where that protein will produced? That protein is produced in the ribosomes. Okay, ribosomes will produce the protein. We understood that. Okay, but how that protein will change it into that particular shape and how the protein will come to that plasma membrane, attach it there, that's actually the functioning of the endoplasmic reticulum and Golgi. Okay, endoplasmic reticulum will help in shaping of the protein. Okay, once ribosome create the protein, endoplasmic reticulum take within its tubule or vesicle or cisternae and it will modify it. Okay, and when it come to the Golgi apparatus, and it will do the finalization. Okay, finalization of its structure. You will see the structure in biomolecule. Okay, so and then Golgi will cover this in a vesicle and it will send it to that particular location where it's actually needed. Okay, it may be the membrane of the plasma membrane, or it may be the membrane of the mitochondria, or it may be the membrane of a vacuole, or it may be the membrane of the chloroplast. It depends. Okay, where it's needed, there it's sending. Okay. Not only that, we also reading about the phospholipids in the plasma membrane, right? How the phospholipids are created then. All those process, and we also read about this, right? There are like what's that? Glycoproteins will be present, which act as a receptors, we told, right? How the glycoproteins are produced then? All those molecules or all those biomolecules will be produced or finalized in the Golgi apparatus, and it also has the function of sending these things to the next places. Okay, to the next places. What is vesicles? A good question. Vesicles are these kind of structure. We saw in endoplasmic reticulum, right? These kind of circular kind of structures, we call it as the vesicles. So other two are the this one, cisternae and the tubules. Okay, it depends on their structure. We gave the different names. So remember, okay, the most important thing we can see is two things only. Okay, so what are those two things? The two things will be glycosylation, or giving glycoproteins and modifying glycoproteins and glycolipids, and the next thing will be it's actually act as the carrier molecule, which actually send the different molecules to different places. Okay, and there are two phases are there. One is the cis and trans, which actually one is the initiating phase, another one is the maturing phase. Okay, so I just want to add, someone asked about a 3D structure. I added this one because even though in electronic microscope you can't see it properly in a proper way, that's why I changed the structure. Sorry, I changed this one. Okay. Hi Bikash, probably it's your first class or you came to the class, didn't put a message. Okay, next one is a libosome. Like yeah, next to the Golgi is the libosome. Okay, so what is lysosome? Does lysosome have any common name? Do you guys think? So we have a common name for the mitochondria. We have a common name for the this one, like chloroplast. What about this one? Exactly. These are called. Like someone put a message. I didn't. So it's actually scrolled away. Okay, I just put a message. Something about cis and trans phase. Yes, exactly. Suicidal bags. Why it is called as suicidal bags? No. Cis and trans phase not in the same place. Look here. So which one is looking like a concave? This one, right? This one is the trans phase, and which one is looking like a almost like it's not actually convex. Okay, so kind of a convex, right? So it's looking like flat. Still, it's a cis phase. Okay, so this one is attached to initiating to the endoplasmic reticulum from here. Endoplasmic reticulum is connected to here like this. Okay. Okay. Yeah, no, good answers. Okay, exactly. Lysosomes are single membrane systems or single membrane organelles exactly. And these contain very low pH. Low pH means what? Acidic kind of the media, and several lysing enzymes. Okay, lysing enzymes. You can call it, okay, exactly, hydrolytic enzymes. So what's the meaning of lysing then? What's the meaning of lysing? Lysing means breaking down. Okay, lysing means breaking down. So it has several enzymes which actually function as a breaking down material. Okay, if you take a lysosome and if you put those material on anything, this will lyse those things. Okay, what's the function of this then? These have function in two things importantly. One is the internal metabolism, or you can say, or internal digestion, or intra digestion, you can say. Okay, we write it like that only. Intracellular digestion, and it also help in the defense and removal of extra materials, removal of the derpes, we can say. Okay, so what are these things then? So once anything is present near to the cell, what it will do, if it's a food source, okay, think that some protist is there. Okay, take a protist example, and near protista bacteria is there. Then what the protista will do? The protist will engulf itself, right? Engulf the bacteria, not itself, engulf the bacteria. Then this lysosome to go and join to that. I'm just writing a diagram. Okay, think about this one. What is the structure? Can anybody recognize this? Okay, so think that here one bacteria is there. Okay, a small bacteria. Okay, then what happens? What is this? Not Chlamydomonas. Euglena, not Chlamydomonas. Okay, don't say in the Chlamydomonas. It's like I'm writing. I'm telling you guys, it's eating a bacteria. Okay, don't say Chlamydomonas, because why I can't say Chlamydomonas? Why I can't say Chlamydomonas is eating the bacteria? Why? Why should I not say? Should not say? Hi Tejashree, you came here. Why I should not say that's eating that one? Correct, right? Chlamydomonas is photosynthetic, autotropic, exactly. That's fine. So think that it's yeah. So think it's eating this one. Okay. So when what happens, it will engulf this, right? It will engulf this. So it will engulf somewhere like this. Engulf somewhere like this. Then what happens? This lysosomes, think that this is a lysosome present in here. Okay, this is the lysosome. This lysosome will travel from here to here and mix into this. Okay, it will mix into this because lysosome have all the necessary things. But starting an engulfment or phagocytosis happened, right? There's only the membranes of the plasma membrane structures from there, right? This lysosome will give all these materials to this particular engulfed material. Okay, so I'm writing the third diagram here. Then what happens? Then all these materials which is present here, which will started to get destroyed because of the acidic nature or nature of that particular substances in the lysosomes. Okay, then after the breakdown, those nutrition will be absorbed by the plant cell. Okay, nutrition will be absorbed by the plant cells, and extra materials will be there, right? Extra materials will be ejected out, right? Extra materials will be ejected out. So through the exocytosis. Okay, what is that removal? Exocytosis. Starting is endocytosis. This is the endocytosis. This is the exocytosis. That means removal of something from the cell. Okay, so no, it is not a plant cell. I'm telling about a euglena which is eating a bacteria. Okay, euglena eating a bacteria. Okay, so what is happening? Euglena is eat something, then this lysosome go and Okay. Not plant cell. Okay, not plant cell. So that lysosomes which is present in the euglena or a protista go and join to that remove defense removal of what? Removal of derbis means waste. I will come to that. Okay, so it will digest that particular one, and the lysosomes will be separated. Okay, called as late ribosomes and all the things. The waste material will be exocytosis. Okay, so that means intracellular digestion is happening through the lysosomes. Okay, and another one thing is like a defense. So lysosomes also help in a defense mechanism. How it is then? Exactly same situation, but take it something like the bacteria is actually pathogen which is invading your cell. Okay, it is invading your body. Then what our cells will do? Our WBC will go there and it will phagocytosis the bacteria, right? Then what process will happen? Exactly same process will happen, but the reason is different, right? In the protista, it's for the intracellular digestion. For here in our body, it's not for the digestion process, but it's for the removal or for the defense mechanism. Okay, and it can be also used in the removal of the derbis. Sometimes what happens, the cell may accumulate something, so in that time the lysosomes can go there and it can destroy that particular organic matter, can remove the things. Okay, hydrolytic or hydrolyzing means what? It breaking the bonds. Okay, breaking the bonds, that means destroying. Okay, hydrolytic enzymes means those enzymes will break the bonds. That's why the matter or things will be destroyed, get destroyed. Okay, so remember guys, here the lysosomes got the suicidal bags. Why? Because these have the enzymes. Derbis. Sorry. Okay, third and fourth, nutrition will be absorbed. One second. I will write down. Nutrition will be absorbed. I use the same color. I will remove the color so you guys can understand properly. Again, this form the here. Okay, there is no change in this thing. Nucleus is intact, and exocytosis, I will show it in yellow color. Exocytosis. Okay, derbis. I got confused, Lipika. Just check once. Let me know. Okay, I got confused. Okay, we'll see which one it is. So how can remember all that much stuff, biological words and name? Simple, bro. Okay, how it is simple, like imagination first thing and reading. Okay, so you have to read it and after some time you have to revise the things. When you come across with the same times too many times, you will get it easy. Okay, debris. Thank you. So that's the only way. And try to get confused. Use frequently. That's also. I get confused. Leave it like that only. Totally waste materials. Okay, it will remove the waste materials. We'll see that what spelling or anything. Okay. So, what I'm telling you, a story, guys. I forgot. Okay. Simple. We normally call it as the suicidal bags because it have all the hydrolytic enzymes. And when that hydrolytic enzymes, if something, a lysosome may burst, then if it is released to the living cell cytoplasm, then we think that it will destroy all the cellular organelles. So it will be death of the cell. Okay. This way we gave the name lysosomes also. But recent studies show that this is not happens like that because those enzymes are only become active in a low pH. That means pH below 5. Okay, pH below 5 means more acidic, right? But what is the pH of the cytoplasm? The pH of the cytoplasm is almost like 7.2 to 7.4. Okay, so some people will say that even though if it is burst on the cell, it do not cause any harm because those enzymes are not at all active and the medium is also not acidic all the time. Because not always hydrogen ion concentration is that much good inside the things. So cell, nothing will happen to cell. They will say like that also. but it's an old thing. We actually name it in the first place, so that's why we keep it like that. Almost human pH. I'm talking about regular cell pH. Regular cell pH only 7.2 to 7.4 in all the cells. Exceptions are there. We are not going to that. Okay. So, we will see. Last one is a vacuole. This is the last, what we can say, component of the endomembrane system. Vacuoles are the bags which contain all the necessary things to the particular organism. Okay, these are present in the plant cells and normally won't present in the animal cells. Okay, these are surrounded by a layer, surrounded by a membrane. What the membrane is called? Surrounded by membrane. The membrane is called as, one second, I change the color. Yeah. Can anybody? Exactly. Tonoplast. Correct. Okay. We not call it as like plasma membrane or something. Okay. Even though composition is almost same, we call it with a different name, this tonoplast. Okay. Why we call it as the endomembrane? Because these tonoplast layers also originated from the endoplasmic reticulum only. Okay. There only all those things are created. Golgi and endoplasmic reticulum, that's actually created the vacuole. Okay. This vacuole contain normally high concentration of material solutes. Okay, high concentration of solutes. That means even though in the cell the pH will be 7.2 and 7.4, and outside of the cell it may be like same, it may be water or something, but inside the vacuole the pH is completely vary because it will do active transport. You know, right? Active transport can be go against the this one also, concentration gradient. So using active transport, going against the equilibrium or going against the concentration gradient and using active transport, it will deposit the solutes. Okay, what's the use then? What's the function or use then? The use is mainly as some of these act like a food storage. Okay, and some of these act like a excretory storage. Okay, you remember the plants do not have any facility to excrete any waste, right? Excretion, storage of the food or storage of the excretion or storage of the insecticide or toxic substances. We can call it as like toxic substances. Okay, what are these toxic substances then? You know, some of the plants are poisonous, right? Their leaves are poisonous or their stem are poisonous. What is the reason? The most common reason why plants are poisonous, because to stop the herbivory. Okay, herbivory means herbivorous plants will eat the plant, right? To stop foraging or to stop wasting or like losing its leaves and plant structures, it normally have the ability to produce the toxins. Okay, so these toxins are harmful to the plants also. It is harmful to the organism. It may harmful to the plants also. So what these plants will do, the produced chemicals or toxins will be stored within the vacuole. That's why it will be secured. There will be no issue. When a particular organism eat that and when it's chewed it or digested it, then the toxins will be released and that particular animal will be get affected. Okay, suicide planta. It's not a suicide. For this one, toxic, the vacuole containing toxic substance, you can call it like suicide. Okay, but other things are other organisms are dying, right? You can say that like murder plant. Okay, something like that. That will be more better. Correct. It's a murder, not a suicide, right? So it can contain that kind of toxic substance or excretory substances or food storage. It may contain the glucose granules, or it may contain starch or this one, glycogen, all those things. Okay. If the plant will die, that cell will die, not the complete plant, the cell will die. Okay. So that happens because cell is bounded by the structure, right? Those particular structures may start to die. That will happen. And not all the structures will be toxic to the plant. Okay, only few of them are toxic, but the major issue it will cause is like it will vary the this one, the solute concentration of the cytoplasm. when you added too much of the solute, then that will be varied, right? By touching that plant, they leads to rashes. Some of that also there. We will think that we actually only touched the plant, but the issue is like it have a fine needle like threads, right? Needle like trichomes we call it, okay? Stem hairs. Those stem hairs will be penetrated your skin without you knowing, okay? You simply thought that I touched and took back, but it's already penetrated your hand and it's already started reaction. That's also happened. What is sap? Sap means how can I put it? The liquid which is present in the this plant. Okay, liquid. I can call it as liquid. But when you when you are learning about xylem and phloem, that time sap means the liquid present in the phloem, but regularly a liquid. Sir, why vacuoles are at center? No, it never in the center. Okay, so normally what happens? The vacuoles will be increased in such a way that it will push all the organelles to the side, so it will become central. Okay, so it's not always central. It will push the other organelle, so that's why it will become the central. There is good question actually. It reminded me another one thing, another fun function. Okay, so another one function is this vacuoles. By pushing the organelle, it helps these plant cells. How it is? By pushing the organelle, it's actually pushing the chloroplast to the outer side, where actually it will get the sunlight. Okay, where it will get the sunlight. Okay, what happens if the mitochondria is somewhere here, nucleus, vacuole is somewhere here and here, then the sunlight which is entering the chloroplast will be decreased, right? But because these chloroplasts are towards this end, it will get lot of energy. That's also there. Okay, that's also one of the function of the vacuole. Okay, so these are the major things which related to the vacuoles. So okay, we will. I forgot another one thing. Okay, so another thing is like in the sum of things we also seen the food vacuoles. Sorry, not food vacuoles, contractile vacuoles. Okay, what are contractile vacuoles? Contractile vacuoles are normally filled with a gas or any other substances. These are help in the like floating, that means buoyancy. Okay, and it's also help in the movement of the organism by pushing the cytoplasm in different way. Okay, why this two growth happens? Like why? Because it simple. Not all cells will contain this much of large vacuoles. Okay, when more storage organ will start to accumulate in the inside, it need more space, right? So it will increase its size. So larger materials, larger vacuoles. So if you see some storage, if you take potato, cell of the potato, you see a large vacuole and very few of the organelles. Okay, if you take the same some stem or some other plant cells, then vacuole will be comparatively smaller. So it's always depending upon its function. Because what is food vacuole? The vacuole which contain the food or food storage, is doing the food storage, is the food vacuole. Do aquatic animals have contractile vacuoles to regulate buoyancy? Aquatic animals don't have, but protistants have. Okay, aquatic animals will have the either this one, what we can call the fishes will have those structures, right? A structures and all those things. Single cells normally don't have the vacuoles. Air sacs, sorry. Push from the vacuoles do growth happens. Push from the vacuoles to growth happens. Growth is a complete thing, not only by the pushing of the vacuoles. Pushing of the vacuole may increase the cell surface, but it's like overall growth. Amoeba have, exactly. One question came, why such large as it is? Because it having more compounds. When it's fully or pushing with more compounds, it will become more and more larger. Okay, if you put more oil droplets into that, it will become much larger. Besides producing secretory vesicles, the function of Golgi body is lysosome formation, formation of spindle fiber, formation of endoplasmic reticulum, all of the above. I'm just launching the poll. Sorry, I'm relaunching the poll. Okay, Manisha. Once we complete this question, we will go back to that. Okay. Okay, I'm just waiting like last 30 seconds. You guys have 30 seconds, okay? Not last 30 seconds. Correct. Okay, I'm ending the poll in 10 seconds. I'm ending the poll. Yes. And I'm sharing the results. 50% chosen option A, 10% B, 14% C, 26% D. Okay, we'll see which is the correct answer. So, besides producing secretory vesicle, the function of Golgi body they are asking. Okay, function of the Golgi body. Formation of the endoplasmic reticulum. No, it's a vice versa, right? So, endoplasmic reticulum will give rise to the Golgi body, not Golgi body give rise to the endoplasmic reticulum. It's a completely ulta. So when we delete the third option, we can also delete the fourth option because all of the above, but third is wrong. Formation of the spindle fibers. No, formation of the spindle fibers will be done by the other one organelle. We call it as the centrosome. Okay, centriole or centrosome. You can call this. So this is also not true. The correct answer is lysosome formation. The Golgi body vesicles may give rise to the lysosomes. Okay, vesicles in here will turn into the lysosome. Okay, so yeah, somebody is asking about contractile vacuoles, how it will happen. Okay, I will give the answer. We will take euglena only. Okay, euglena is the best answer for that because euglena contain a contractile vacuole. Okay, think that where it is present, the complete this one water will be or where it is present is actually hypotonic solution. Hypotonic. What is the hypotonic? That means water will be having less this one ion concentration than the inside the body. Then what happens? All the water will start to move to the inside, right? All the water will start to move to the inside because salt concentration high in here and low in the outside. It's moving from higher concentration to the lower concentration. Water is moving to make it a balance. Okay, because solids can't move in that way. Okay, sorry. So what happens? We'll see. So in that time, what happens? This vacuoles will get filled with the water. Okay, this vacuole get filled with the water and collect all the water which is present in here. Collect all the water, and while moving on, it will pinch off this water to the outside. It will pinch off the water to the outside. This is how it's actually maintaining its osmoregulation or the ion concentration. Okay. Even in the same way, it will also do the function for the excretion. Okay, it will collect excretory substances, which is actually produced by engulfing some materials. Okay, and it will come and attach to the plasma membrane, and it will pinch off or excrete all the waste to the outside. Okay, amoeba. This one you can take all those examples. Yeah. Okay, so we'll move on. Prevents bursting. Correct, actually. Prevent bursting. That's also you can see. Next one is the mitochondria. Mitochondria is one of the cell organelle which have the double membrane system. Double membrane system. Okay. What is the meaning of double membrane system? It have two membranes. Until what we see, everything had a single membrane, right? But this one have the two membranes. One is the outer membrane, another one is the inner membrane, as you can observe in this portion, right? Outer membrane and the inner membrane. Okay. So what it will do then? How it will get the double membrane? So, and another one thing I will mention before I'm telling how it get double membrane system. That is these are the one of the organelle which having the DNA in the form of, or you can call it as circular DNA. It have the circular DNA. You can call this as the plasmid also. Okay, it have its own DNA, right? And what are the other characters? This have the 70S ribosome, right? These are the major three characters what you can observe. And how we call it as? We call this as the powerhouse of the cell. Powerhouse of the cell. Okay. So now probably you guys guessed right how it will be different from the other organelles, because this have the double membranes and it have the circular DNA, and it also have 70S ribosomes. Okay, it's like completely different from the other organelles. So we leave the chloroplast for now on. Sorry, for this segment. Okay, so how we got this? It got all these things because it's a endosymbiont. Okay, we got it. So, why do we have the 70 ribosome? Why do we have the DNA? Why do we have the double membrane system? Because none of the other organelle have. Because this mitochondria came into the eukaryotic cell as an endosymbiont. What's the meaning of endosymbiont then? This mitochondria is somewhat like this one time. Okay, this is mitochondria, this is its chromosome. Okay, so chromosome. Okay, this is actually a bacteria. Okay, this is actually a bacteria, a kind of a parasitic, we can say, one time probably. Okay, then what happened? There is a kind of protista we can say, or eukaryotic cell, think something like that, which having the nucleus and the nuclear membrane. What it did, it actually did the phagocytosis kind of the nature. Then what happens? It leads to the, it leads to this eukaryotic cell something like this. It engulfed the thing. When it will engulf, then what happens? It becomes something like this. Understand? So this eukaryotic cell actually engulfed this bacteria, and what this eukaryotic cell did is like, it haven't destroyed that bacteria, it actually kept the bacteria. Okay, when it kept the bacteria, then what happened? They build up a relationship. Okay, what kind of relationship? The eukaryotic cell told, look, you don't need to do anything, I will provide all the basic material what you need. Okay, but you have to give me the energy. Bacteria told, okay, like, because otherwise it have to move away, move here and there and collect the materials and create the energy. But it's like simple, right? It should sit somewhere, and everything will be, raw materials will be come up to that, and it have to like create that into the energy molecules and use it for itself and give it to the cell also. That's a good thing then. So it started doing that. Now probably you can understand why it had the 70S ribosome, right? Because it's formerly a bacteria only. Bacteria will have the 70S ribosome, so not the eukaryotic members, right? So that's why mitochondria will have the 70S ribosome. Why it have a circular DNA? Because circular DNA only present in the bacteria. Because this is one time a bacteria. Why it have a double membrane system? In the double membrane system, as you can see, the outer membrane, what you can see now, is actually membrane of the host, right? Host membrane, when it engulfed it, as you can see, white layer, right? It's the outer membrane. The inner membrane is the true membrane of itself, true membrane of that bacteria. Understood? So that's why it have the double membrane system. Otherwise, it do not have any double membrane. It also a single membrane only. If it's a like regular organelle, it's like simple only, right? So that's why it become all like this. Okay. So any more questions related to its origin? Okay, we will move on to the next then. When you see outer membrane, I will write down a little bit below. Okay, outer membrane. So it's almost, you can like selectively permeable, sorry, semi-permeable. Okay, semi-permeable, or you can say like selectively permeable also. Okay, and if you see at the inner membrane, so it's highly selective layer. Okay, it not at all allow any molecule, only few molecules it will allow inside, all the other molecules it will kept outside only. Okay, it will not allow everything to the inside. Okay, in between the outer layer and inner layer, the space is there. Okay, a space is there, right? This particular space, in between space, you call it as inner membrane space, or you can also call it perimitochondrial space. Intermembrane space, inner membrane space, intermembrane space, perimitochondrial space, intermembrane space, or we also call it a perimitochondrial or perichondrial space. Okay, mitochondria, so chondra, like that. Okay, perichondrial space. Okay, or you can call it as intermembrane system. This is the important place. Okay, don't think that the important place in between. Actually, the important space is this intermembrane space. That is what the pH or the ion concentration here only actually matters for the production of the ATP. Okay, and next, if you see, or if you see closely, you can also observe the inner membrane is highly folded. Okay, this folding nature will give rise to a finger-like projections. What we call that one, the finger-like projection, which is created in the inner membrane of the mitochondria. No, not Priyanka, not that one. I will tell you. Okay, called cristae. Exactly, it's called as crista or cristae, singular or plural. Okay, cristae. Okay, this cristae is called, cristae are the finger-like projections. Finger-like projections, these are called cristae. Okay, it's like finger-like. Why it is doing that? What is the reason for that? Because the inner membrane is folded to increase the surface area. Because of its folding, the surface area is increased several folds, right? Instead of having a single layer, if it do the foldings within a minimal volume, it can, minimal area, it increase its surface, right? It can increase its surface area. So cristae are finger-like projections created to increase the surface area. This cristae will, exactly, villi in intestine. Okay, sorry. So I will write something like this. Okay, I can write something like this. This cristae will have certain structure on them. Those structures are called as F0, F1 particles, or it's also called as oxisomes. Okay, what are oxisomes or F0, F1 particles? F0, F1 particle or oxisomes are similar. Okay, how can I say? These are also almost like finger-like only. Okay, structures on surface of, inner surface of cristae. Okay, this cristae will have F0, F1 particle. If I write a diagram here, 1 second, I will write it properly. Okay, if I write a cristae, Fernandez Moran particle, you can call it in different names. Okay, so we will write something like that. This will be the F0, F1 particles. Okay, so these are the F0, F1 particles. What are these F0, F1 particles? You can also call it by the name of oxisomes. STR means structures. Remember, it's a short form. Okay, so several times I will write like that. Structures. Don't write it in exams. For the notes you can use. Okay, STR means structures. So what is the use of these oxisomes or F0, F1 particles? These are the structures which actually produce the ATP. ATP production will happen through this one only. Okay, how it will happen, all those things we will learn later. Okay, there is like a gradient of the hydrogen ion or protons. While moving of the hydrogen ion, we will get the ATP molecules. Okay, so we learn how, and the thesis, like hypothesis, chemoosmotic hypothesis, everything in the later part. For now, you can remember plasma membrane is made of finger-like projections. We call it as cristae, and this cristae will contain another structures on their sides. We call it as F0, F1 particles, or you can call it as oxisomes. Okay, the function of oxisomes or F0, F1 particle is the ATP production. Okay, these are related to the mitochondria. Okay, so, and there are some other questions we encountered with. So what is the function of the DNA in there? right? Why it should contain a DNA? This DNA also code for certain genes, so this will also help in the protein synthesis. Okay, the protein which are needed inside the mitochondria are produced from its own, some of the proteins are produced from its own DNA. So this DNA will produce or code for RNA, that RNA will go for the protein synthesis. This 70S ribosomes will help in this protein synthesis process. Okay. Someone asked the question, right, where actually protein synthesis happens. So happens in the matrix. Sorry, I forgot the thing. So what is that? Inner membrane contain a fluid-like structure. That's why I call it as the matrix. Like the cytoplasm, it is the matrix. Okay. How mitochondria gets divided? That's a good question. Okay. I told you that it's a different cell organelle. It's a bacteria. Then you could have guessed right. How bacteria will divide? How bacteria will divide? Exactly, by fission. So same thing happens. Mitochondria never follow what cell is saying. Okay, when it feels that it should get divided, it will divide. Okay, when it feels that like lot more is there, then some of them will get destroyed. Okay, conjugation, we normally won't see the conjugation in mitochondria, but we definitely see the this one only. Okay, fission only. Okay, probably it lost all those ability. For the conjugation, all the process, it need plasmid, right? It have a single DNA, that's also circular DNA, so it do not have an extra plasmid, so probably it do not have those works. As we've seen, there is no conjugation, but it show the bacterial kind of the binary fission. Okay, so where it will do, simply think about it. If you provide more energy to a single cell, then the bacteria will divide, that mitochondria will divide, because it need to process a lot of energy, right? So same kind. And the number of mitochondria will be vary from cell to cell. If you take a simple, our epithelial cell, the mitochondria will be less. If you take a muscle cell, then the mitochondria will be more, because muscle cells need a lot of energy, right? To produce that, it will be more in those kind of cells. Okay, isn't sir F0 F1 particles are like particle in chloroplast, elephant membrane? Exactly, Akshay. So if you already learned chemoosmotic hypothesis in the plant physiology, there is also F0 F1 particle. So those and here are same. Why it is same? Because F0 F1 particles are nothing but these are proton channels, you can say. Okay, those are proton channels. The name is F0 F1. We call it as like F0 F1 type of the proton channels. Okay, that is not in the syllabus. So both are same, you can think. How it help in the aerobic respiration? That's what we will learn in the photosynthesis, because it's a long form. I will tell you. And like crystase this one. Remember, crystase this one. Crystase is finger-like projections. Inside to this vacant space is there, right? There actually matrix is present. Okay. Porins are present. That's what make outer membrane selectively permeable, because porins are present here. Outside membrane, not inner. In outer membranes, porins are there. Okay. How it will do the aerobic respiration means they will take certain materials or the basic compounds. You will see what are the basic compounds and everything. Then there are some proton channels will move through this cell, move through this particular lines, and they will create a gradient of the proton. Okay, proton means hydrogen, this one, ion, right? So it will create a proton gradient. When breaking the proton gradient, the protons will move from one higher concentration to the lower concentration. By utilizing that energy, the moving energy of the proton, it will create the ATP. Okay, we will see how it will happen in the physiology. No issue. Okay, so you can think by creating the proton gradient in the this one pericardial, or intermembrane space to the inner. Okay. Is then are F0 F1 particles are proteins? Yes, F0 F1 particles are proteins only. It's not a single proteins. It's actually combinations of 5 different kinds of proteins. And it's a big structure. Okay, we will see structure and everything. It's a big structure. It's like your, how can I say, this one, guys, turbine, right? Water will come from one side and turbine will rotate and it will give the electricity, right? Exactly same kind. Proton will pass from one side to another and it will turn the wheel or proteins. Okay, when protein is turned and it go for some conformational changes and create
it will join adenosine diphosphate to another one phosphate group, and it will create the ATP. If you don't give a lot of stress, that we will see that. Okay, I know some people are not at all understanding what I'm saying because you haven't read the plant physiology. Okay, no problem, I will explain that later. Okay. Next one is the plastid. But think about like a turbine, that one, that will be good. Okay, so what are plastids then? So what are plastids? Plastids, not plasmids. Okay, plastids are pigment containing double membrane bound organelle, and these two also have circular DNA. And what of ribosomes? If you go for ribosome, it have the 70S ribosome. Okay, so all is left to you guys. What do you guys think about the plastids then? What do you guys think about the plastids? So I gave these some characters: circular DNA, 70S ribosome, pigment containing double membrane bound organelles. Correct, Lipika? Anybody else like? Exactly, right. This is also a bacteria. This is also endosymbiosis. It's actually the same kind, same story like the mitochondria. Okay, the only difference is this is not actually simple bacteria. This is actually a cyanobacteria. Okay, cyanobacteria, you know cyanobacteria, right? Don't think that that same cyanobacteria is chloroplast now, because in the millions of years, lot of changes happened. Okay, all these things happened billion years ago. Okay, remember what I'm telling the story, right? This story happened billion years ago. Exactly, Sachin. Okay, this story of the plastid and story of the mitochondria happened billion years ago. That's why don't expect the same thing now. Okay, don't expect the same thing. The changes happened here. Exactly. This is also a bacteria, but it is not like a simple bacteria. It's actually a cyanobacteria. Okay, once upon a billion years, cyanobacteria. Okay, so same thing happened. The one eukaryotic cell went and actually took this one inside. How they came to know this exactly this happened? I will tell you that. Okay, so it also took back this one cyanobacteria, and that's why it have a two membrane. One is actually plasma membrane of the host, and another one of the membrane of the chloroplast. Okay, you know, when you go for cyanobacteria, it should have another one membrane, right? You should have a cell wall. Exactly, right? Where the cell wall went, cell wall got removed. Okay, cell wall got removed in the process because cell wall is needed for the mechanical damage, right? But here there is no mechanical damage because it's already surrounded by the complete up another plasma membrane and all the fluids. It's already present in another one, right? So that's what happened there. It lost its cell wall. Okay, it made certain changes in itself, so it's actually working like this. Okay, now the question before we moving on to the secondary things. Okay, how scientists came to know about these things, right? Because in the initial we also thought that these are the original organelles, but we got one question: how the eukaryotes are created. So hemoglobin is plastid. No, hemoglobin is not a plastid. No, remember that is there. That will be there. This will be there. Okay, so pigment is the organelle. Okay, sorry, pigment containing organelle is the plastid. Hemoglobin is not a plastid. Okay, I will come to that. What happened, like when we have that question, we will try to find where it is created, how it is created, everything. Okay, when we go through the DNA and other components of the chloroplast and the mitochondria, we struck with one thing, that is these two resembles exact like a common bacteria. Okay, you know that the prokaryotic cell is small and the eukaryotic cell will be larger, right? The size of the mitochondria or the chloroplast or the DNA genetic material or the ribosome or the RNA, okay, everything looks exactly like a bacteria. Okay, exactly like a bacteria. Then we actually sequenced the things, sequenced the DNA, and we did a comparison. Okay, then we came to know that these are nearly like this. Probably this is what happened. Okay, Does that all double membrane organelles are bacteria? The thing is like only two double membrane organelles you can find. One is the chloroplast, another one is the mitochondria. So you can say that. Okay. So then they hypothesized like this. Probably this is what happened several billion years ago. Okay. So there is another one thing is there. That is how they interact with the cell. Okay. How they interact with the nuclear DNA. Okay. So how it will get the proteins, or what it's like you will learn in 12th class. Okay, some of the things, how it is coding the protein. Okay, what kind of codons it is using, what kind of tRNA, rRNA, mRNA is present. Everything is we started working on. Okay, so and there is actually one this one, when you go for mitochondria, there is one bacteria is there. I actually forgot its name. Okay, it's actually a parasite of the eukaryotic cells. Okay, parasite of the eukaryotic cell. Remember, not the endosymbiont. It's a parasite. Okay, parasite of the other cells. It looks exactly like a mitochondria. Okay, that particular parasite exactly looks like a mitochondria. Then we come to know that okay, it may have a similar origin. Like okay, so that is how we discovered the stories and all the things. Okay. So plastids are different. Okay, there are like several structures are there, but the simple thing, as you can say, is outer membrane. It have a two membranes. Outer membrane is normally semi-permeable or selectively permeable. Semi-permeable, or sometime this is semi-permeable to the permeable, like cell wall only. Okay, and inner membrane is highly selective. Inner membrane selective. Okay, it's a selective membrane, and not all the things which is present in the chloroplast are secreted by the chloroplast only, like that of mitochondria. Here nucleus, or we can say nucleus, play important role in maintenance of chloroplast. Maintenance of chloroplast. How that will be? That means this some of the proteins will be coded by the chloroplast, and some of the proteins will be coded by the nuclear or genomic DNA, and that particular RNA or protein will be transferred to the chloroplast. Then chloroplast utilize that RNA or DNA, RNA or that particular protein as it needed. Okay, so that is how happened. That means mitochondria is almost like independent, but chloroplast need these things. Okay, what is plastome? I don't know, Mohan. What's the difference between semi-permeable and selectively permeable? Good question, Jayant. Semi-permeable means it only allows some of the particles, but it never selects those particles. Okay, think that membrane is there, it will pass only positive ions. Okay, then you can call it a semi-permeable because it's only passing positive, not the negative, right? But a selectively permeable membranes only pass a selective ions. Like even though it's a positive, it will select the positive ion. The membrane only pass H plus ions, protons only, may not pass the Na plus or K plus. Right? Then it will become selectable. Semi-permeable pass all the kind of things, but selectively permeable select the particular things, then only it will allow to pass. Okay, even though some same structure, different compound is there, it won't do that. Okay, we have some carrier proteins. Okay, carrier proteins. That carrier protein we call it as Na plus carrier protein. Why? Because it will only pass the sodium ions, not the potassium ions, not the protons. Okay, difference between plastids and chloroplast. I will come to that. Okay, that's the difference. So types of plastid. We came to here. Okay. So when we come to the types of the plastids, there are like some types are here. Okay. So what are those types then? What are the, one second guys. So what are the types then? The plastids are divided based on its function and what are the character it have. Okay. First one is the etioplast. These are crystalline prolamellar bodies. Prolamellar means behind to the lamella or creating the membranous structures body. Okay, This is what divide to form the chloroplast. Okay, where you can find the etioplast. If you've seen any germinating seed, okay, initially it will be white in color, right? Later it will form the green when it is like encounter with the sunlight. Okay, you make an experiment. You grow some of the seeds without light condition. That means completely in darkness. Okay, the plants will grow up to some height. Late, but if you see the plants, everything will be white. Okay, why? It do not have the chloroplast. Chloroplast only develops when it will hit by the sunlight. Until that, it will be have the etioplast. Okay, this etioplast when get this one sunlight will convert into chloroplast. Until that, it will be having a crystalline kind of a body like structures. Okay, that's the etioplast. Probably this one I think you are asking. Okay, next one is the chromoplast. Okay, chromoplast is a generalized thing which now actually synthesis pigments and do the storage. It normally contain keratin, okay, or xanthophyll. Okay, so this is the reason why you will see a different colors of leaves. Okay, if you go for a winter season, you see that the color of the leaves will changing, right? Why it's happening? Because the chloroplast are converting into chromoplast. Okay, or sometimes chloroplast is decreasing. That's why the leaves are turning from green color to the orange color or red color, or some is like yellowish color, different colors based on the different pigments which is present in that particular plastid. Okay, that's what we call it as chromoplast. Chromo means color, right? Chromo means color. Next one is a chloroplast. Chloroplast which is doing the photosynthesis, okay, and also do the lipid synthesis. Major is the photosynthesis, what we are encountering in next slide. Okay, so normally present in the plants. Next one is a leucoplast. Okay, leucoplast is a kind of plastid actually doing the fatty acid and amino acid synthesis and accumulation. Okay, normally it will do the fatty acid and amino acid synthesis. Okay, so just imagine about etio and leuco. Etio will convert into chloroplast. This leucoplast may convert into some these types. Okay, etioplast may convert into these two types, and leucoplast may convert into these three types. One may be amyloplast. Amylo means starch. Okay, that means you can simply think about that. Okay, amylase, you know, right? Amylase which is present in the saliva. That means the enzyme which actually destroyed the starch. Exactly, amyloplast will store the starch and the sugars. Okay, and elaioplast it actually store the lipids and oils. And proteinoplast, so it actually do the site for the enzyme, especially a protein synthesis or the accumulation, you can see. Okay, so these are the three things which are important with regarding to this. Okay, and I think I left one other thing. The proteinoplast may also called as alluroplast. Okay, alluroplast. Both of the things are same. Alluroplast or proteinoplast, both of them are help in the site of the enzyme synthesis. Okay, one second, guys. Okay, so these are the major things which are doing this one. Okay, so next one will be the chloroplast. We will see about the chloroplast. Okay, what the chloroplast will do and how it will work, everything we will see that in the next class. Next class means Tuesday. Okay, so I launched the teacher NPS. You can like answer the teacher NPS, then you can leave the class. If you have any doubt, you can ask now. Okay, even in the YouTube also, if you have any doubt, you can ask now. And probably this one, Akshay, you misunderstood this etioplast. Just check with that once. Okay, so if it is ectioplast only, just let me know, so I will like try to find. Chloroplast convert into chromoplast. Exactly, chloroplast may convert into chromoplast in fire or flame. What fire or flame, Darshan? I didn't understand. In our TH also is our THS also result of excretion process? No, actually, kind of. Okay, There are like substance, like we have some glands here which actually keep the ice moistened, because ice is an internal body structure. It's actually exposed to outside. It can become dry. When cells become dry, it will die, right? So that's why a process is there. But when you are experiencing some chemical or any substances on your eye, our eye have to get, I want to get rid of that, because none of the other things can help that, right? If something get into your nose, you will sneeze. If something gets into your mouth, you can split, right? If you something stuck in your ear, wax is there. But what is there for the eyes? So that's why they will secrete the tears. These tears can increase that nature, or what you can say, it can remove that derbies from your eyes to the sides. So from there it can be removed. Okay, kind of excretion you can say. So, ha, I remembered about it. Yeah, yeah, most probably, bro, Jivan. Yes, I forgot actually. That's why. Most probably. I think I thought that probably it will contain that. If in is fine. One second, one second. In fire and flame, the chemical composition. No, it's not a chemical. Fire and flame is complete process. It's a oxygen and burning carbons, hydrogen, everything. It's a process and everything. That's why we never put fire and flame in any one. This one. It's not a solid. It's not a liquid. It's not a gas, or it's not the any other thing. It's a complete process of burning. Okay. If injury happens to sugar patient, it will not cure fastly. Why? Because if sugar is in high content, sugar will stop the thrombin molecules to work. The first case. Okay. Second thing, what happens? The same sugar molecule may crystallize the things, and it will allow that bacteria to grow on that. It may cause the gangrene. The first thing why it is not cure is because it's not allowing the pathway of the what is that, clotting pathway to take place. It will stop that clotting pathway. Second, why it cause the gangrene? Because it have the rich sugars which allow the bacteria to grow. So can you once again explain the last class? Okay, I will. Usually contain some compound, bro. I forgot the sulfate compounds. That particular compound actually come and like deposit on our eyes, right? It's a irritation. It causes irritation. That's why. Lysosome. It contains lysosomes, but I forget about I forgot little bit. That's why I'm not sure. I told. The proteinoplast, alluroplast will actually the sites of the enzyme or protein activity. Sometimes it can also show the protein storage. Okay, remember, it is not a site for enzyme or protein production. It's not a site of production. Site of production is ribosome only. Okay, this is just holding that protein. Okay. Yeah. We will put in this way. Hold on. Okay. So in the initial only I told, right, DNA is stable, but RNA is unstable. Okay. So when any substances which come across with the RNA, the nucleotides will get changed. Okay. It have 4 nucleotides. We will see. So when it gets changed, it may get a good chance or it may get a bad chance. Okay. Sometimes due to that mutation or due to the changes, the virus may lose its pathogenicity, or it may lose infecting any other people's. Okay, but those kind of virus will get vanished, unrecognized. Okay, sometime what happens, in a rare instance, it will get higher pathogenicity. That means higher level of causing the this one disease. That time we will get recognized because it's causing a lot of harm, right? That's why a change in DNA sequence, because RNA is unstable. So that's what happened. Thank you. Any more doubts? Central dogma, simple, Akshay. Simple thing like this. Okay, all the DNA replicate and give rise to the DNA. Okay, and the DNA can also transcribe or transcript and can produce the RNA. Okay, DNA can produce the RNA. That RNA by going for the translation and can do the proteins. Okay, so it's like a step. DNA can do the DNA by replication. DNA can produce the mRNA through the process of, RNA through the process of transcription. That RNA in turn can produce the process of translation, can produce the proteins. Okay, that protein will control the total organism. So that's what we call central dogma of biology, central dogma of molecular biology. Okay. Which enzyme will active the lysosomes? No, it's like not activating kind of a thing. Okay, these things will be activated by several submolecular signals. Okay, so those are also protein only, but we are not going to that. Like there are like several kinases and other proteins are there. Those are initiate the lysosomes. Okay, once it's initiated, so it will decrease its pH and its hydrolytic enzymes will work on that. So majorly activation will be based on the protein molecules, not on the enzymes. Difference between plastid and a chloroplast. All plastids are not chloroplast, but all chloroplasts are plastids. Okay, simple. So plastid is a common name given to all the things which are having double membrane and all the characters what we saw. Chloroplast in a kind of a plastid which have a chlorophyll. So that's the difference. Thank you. Bye bye. Okay, guys, then in YouTube, I close the YouTube session. Way to reach me. I'm in the Telegram. So if you have any doubts, you can post it there. Okay. Bye bye.